Hi, chapter 18.4, last topic for genetics. And in this topic, we'll be looking at some more about biotechnology, cloning, and genetic testing. So to start, some transgenic organisms that have DNA from multiple species are used for things like hormone production. We mentioned that one before. So this bacteria carries the genetic code for insulin so that as it reproduces, that insulin is produced. Necessary proteins needed for cancer treatments, necessary proteins that were missing if someone has hemophilia and cannot have their blood clot. So that is a protein that can be made with this biotechnology. And another example is human growth hormone. As you can see, the picture of these brothers, one of them likely needs some of that. The word bioremediation is using cells to improve the environment. So we've got things like plants that take up contaminants from the soil. We've got bacteria that can take up the chemicals that are left over from an oil spill or chemicals from water. So there's using living organisms for useful things. Transgenic plants are genetically modified plants. So that is a lot of the corn and canola in North America. And what they're doing is adding genes that help them resist chemicals that are used to kill weeds, herbicides, resist maybe insects, resist diseases, or even tolerate the cold, which is why tomatoes are given genes from fish so they can survive colder weather. There's always the controversy that comes with genetic modification because it's not a natural sequence. There's research that is being done about whether it is something that nature will enjoy. I don't know, this is a true picture. However, this indicates how the controversy goes, that if you have all natural sequences, it may be more palatable or desirable to organisms in nature, and genetically modified corn, not so much. And so in some countries, this is something that is happening. That's like European countries do not allow genetically modified products in for sale there. They do not sell genetically modified flour or rice or anything, and vegetables, fruits. So that's something that they basically say we'll see how North America does on their genetically modified foods before we decide that we would want to consume them or sell them in the European countries. So something interesting to research. But then the other side of the coin is how scientists who work on genetic modification feel that there are the benefits, like this golden rice is an example where they have modified rice to include iron, vitamin A, and these are other genes that help absorb the iron. And it's one of these situations where they've taken genes from these other plants and added them to the rice, and then they give this rice to people in places that they know are deficient in the iron and vitamin A, so they don't have to change their diet or take vitamins. They just continue eating their rice and they have those supplements in there. So they feel like that is helping humanity with genetic modification there. The gene gun is a technology that can basically zap the DNA sequence that is being put into the whatever DNA that they're modifying. So that's a technology that's used. And they have gold basically that carries the DNA in because it's a little heavier and you can shoot it into a sequence. Okay. And transgenic animals are ones that, for example, we have milk that can carry human proteins that can be given to children who are deficient in or at risk from this certain infection. This was kind of an experiment to see if the goats could have spider silk in their milk if they added the genetic code to the milk, and this was a successful experiment. So they basically put the gene for the spider silk into the milk, and when the goat was creating the milk, there was spider silk in there. And spiders, they don't like to be farmed for their silk, so this was an experiment of how that silk with very high tensile strength could be mass produced, but it was more of an experiment. I don't think they're mass producing spider silk using goats anymore, but it's interesting to read about. Cloning is when many ways to get cloning, but it's identical genetic offspring as the parents. So things like runners, or when we talked about how sponges or yeast can do budding, those are clones as well. We also know that if an egg cell breaks off a cell, like in very early stages, like 8-cell stage, those cells can break apart and become clones of the other cells. Other ways is that we can do cloning from embryos, where we have the case of Dolly the sheep, which was the first cloned mammal. This was done in 1997. So basically in this diagram, you can see what happened. They took one cell of this sheep and they happened to take it from the udder of the cell. And they had another sheep that was donating egg cells. They removed the nuclei from the nucleus from the egg cell. So that would be no more genetic material from this one. And what they did was they took the genetic material out of, which was already a full number, diploid number of chromosomes from this donor, and put that into the empty egg cell. But the egg cell is ready to start dividing. That's what egg cells do. So when this egg cell started dividing, here's a little morula stage. When it started dividing, it was then a clone of this one here, because that's whose material was put in there. This was big news. However, as you can see here, that came with its struggles, because they had to try 277 times before they were able to get a sheep that survived. Also, it only lived 6 years old, and it was basically born as with a cellular age of the older sheep that they cloned. So there is again the controversies that are associated with these technologies, and there is possibly more chance of disease and defects, and of course the premature aging, and so that's the controversy there. Okay, so this is the doctor who was working on the cloning, and that was a photo of the sheep that he cloned. Okay, therapeutic cloning is where stem cells can be used to repair an organ or a tissues. So they could have organs that aren't functioning, and if they could get these stem cells from the person, they could basically take those cells and with the right cocktail, that's what they call it, put these cells in and have them start multiplying to replace that organ. So that would be called therapeutic cloning. So they could, for example, get it to make skin cells or nerve cells, or pancreatic cells to make insulin with using that method. Organogenesis is where they can take cells and theoretically start an organ growing and put it into a, let's say, pig, to have the pig growing those organs for that person to replace organs they're waiting for to transplant in. Really cool video here about growing heart from stem cells. Check that out. So they have a scaffold like that cells can grow onto, and they were able to grow a little heart. So there's lots of future in that there. Controversy here is that with genetic information, who owns that? We have all these technologies, but should an employer have it? Should your mate have it? Can you be tested? Can the judge get access to it? How about the insurance agent? So there's all these questions when technologies come up, and there's the positives of maybe getting your, perfect treatments for disorders, things like that. So sometimes there is, the two sides in all technologies. There's good reasons to do it, and some that maybe are indicating that you are losing your human rights. So also the risks is that there could be genetic modification causing a superbug or superweed that has no predator in nature, and then they can upset the balance and grow uncontrollably. So there's that big concern as well. And to finish up, diagnoses of genetic disorders, ultrasounds are used very commonly to check the unborn babies, often used for other body parts as well. But ultrasounds are just sending sound waves down, and they can see a lot of anything that is like bone and even see heartbeats in ultrasounds. There are now 3D ultrasounds that, like this, that give you a real image of the baby. I'm going to show you this ultrasound because it's special to me. This is my granddaughter that will be born in about 5 weeks. So maybe I'm a little bit excited about this ultrasound photo. And she's already so cute and looks like me. Okay, that'll be my son's first baby. All right, moving on. Second one will be the amniocentesis. That is basically checking fetal cells. So they will take the cells from the amniotic fluid and do a karyotype and check if there is the correct chromosome count in that, because those baby cells will be in the amniotic fluid. Here, the chorionic villi sampling can be done very early on, and basically taking chorionic cells. So they're from the embryo, so they're baby cells, and they will be used as well to make a karyotype. So it's sometimes just abbreviated as CVS, chorionic villi sampling. So both this and the amniocentesis are used to make a karyotype. And then alpha fetoprotein test. This is just to see if there's neural tube defects, because cerebrospinal fluid proteins could be found in the blood sample. This is more of a blood sample test. Okay, so gene therapy is where a correction can be done of the defective gene, and so this could be a cure for genetic disorders if the correction can be put in there. If it's done to body cells, it's somatic gene therapy. If it's done to sperm or egg cells, that would be germline therapy. So if a sperm is carrying a certain maybe deadly gene, that could maybe be replaced before that sperm is used to fertilize the egg in some in vitro fertilization process, perhaps. So gene modification is when that gene is re-modified, so the bases are exchanged, whereas gene surgery, you're putting the entire gene in there to the DNA. Gene insertion would be where we use a vector, as we mentioned, like a bacteria or virus, to send in the appropriate genetic code to do some genetic repair that way. And I guess that about sums it up. Alrighty, so we're gonna wrap up this topic, and thanks for listening to this genetics talk. And I